Very often a question is asked, mention the vitamins whose deficiency may lead to anemia. Among them, first one should always be B6 or pyridoxamine. Second one should be folic acid, and third one should be B12. Any other vitamin whose deficiency may lead to anemia? These three are major. The other vitamin whose deficiency may lead to anemia is vitamin A. So, folic acid, the name is derived from folium, means leaf. That means the source of this particular component of this vitamin is from leafy vegetables. World War II, correct? Otherwise, we cannot perform those experiments. All human rights will stop us. Who will allow a human to suffer from this deficiency knowingly? That to the deficiency of folic acid is very, very dangerous. So now it has three parts. A folic acid has got three parts: a pyridine ring, PABA or para-aminobenzoic acid, and a number of glutamic acid residue. It may be monoglutamate, di, tri, tetra or pentaglutamate. This is a structure. This part is pyridine, this part is PABA, and this part is glutamic acid. Right? Just write down the names and the components. This is important. Why is this important? Why it is important to know the components of structural components of folic acid? This PABA, para-aminobenzoic acid. You found this name in another context, in another chapter. Which chapter? What context? Competitive inhibition of sulfur drugs. Are you remember? Remember, ball.
PABA.
Bacteria synthesize
Folic acid. Using PABA. But sulfur drugs competitively inhibit PABA, so folic acid cannot be synthesized.
Why sulfur drug and how it inhibits synthesis of PABA, synthesis of folic acid? The sulfonic acid is structurally similar to para-aminobenzoic acid. So, during synthesis or biosynthesis of folic acid, it takes up the position of para-aminobenzoic acid, and hence the further synthesis of folic acid is stopped. So, the bacteria dies due to lack of folic acid. Why? Because folic acid is required for biosynthesis of nucleotides, DNA, RNA. Without DNA and RNA, bacteria cannot survive. Right? So, this is the mechanism of action of sulfur drugs or sulfuric acid. Right? So, this is, you should know, of folic acid. There are many coenzyme forms, but mostly they are related to tetrahydrofolate form. To produce tetrahydrofolate, it must go two-step reduction. One is folate reductase, another is dihydrofolate reductase. These two reductase enzymes convert folate to tetrahydrofolate. Clear? Now, tetrahydrofolate may exist in different forms depending on type of reaction they are involved. Right? What are the different forms? Next question. What are the different forms of folic acid? A very your favorite question. What are the different forms of folic acid? Start from N5, end with N5. So, first one is N5 methyl formyl, last is N5 methyl. In between N5 formal THF, N10 formal THF, N5 formimino. Be careful. N5 formimino THF. Then comes N5 N10 methinyl, N5 N10 methylene, methinyl, methylene, and lastly N5 methyl THF. কতগুলো হলো? 6 forms. Let us discuss again. Repeat. N5 formyl, N5 formimino. Sorry, N5 formyl, N10 formyl, N5 formimino, N5 N10 methinyl, N5 N10 methylene, and N5 methyl. এগুলি মনে রাখতে হবে। প্রচুর reactions হচ্ছে, number of reactions are there which require folic acid. The most important vitamin, if you are asked, for human development is folic acid. Now, এখানে দেখো, there are two forms. One is polyglutamate, one is monoglutamate forms. What is the difference? Although they are water-soluble vitamins, a part of water-soluble vitamins can be stored in our body. That is not possible that all the folic acid we are eating today should be utilized by today itself. No. It can be utilized tomorrow, next day morning, day after tomorrow. So that much storage possibility we have. But how do they store? They store it in polyglutamate form. And when they are required to act as a coenzyme, they are converted to monoglutamate form. Not required to know. You don't need to write. The bacterial synthetic biosynthetic pathway, this is the sequence. Sources, write anything and everything which is green in color. Formation of tetrahydrofolate, the active form from folate. Folate reductase, dihydrofolate reductase or tetrahydrofolate reductase. Now these two reductase steps are usually blocked by certain compounds. They are called anti-folate agents and their names are MAT. Bolo? Methotrexate, Aminopterin, Trimethoprim. Remember, don't forget, MAT, Methotrexate, Aminopterin, Trimethoprim. These three compounds are used as anti-cancer drugs. Why? How it is useful to be used as an anti-cancer drug? Anyone? So what? But it blocks the biosynthesis of folic acid, and folic acid biosynthesis takes place in bacteria. So it should kill, the anti-cancer drug should kill bacteria. Why it should kill? It is inhibiting the regeneration process of active form of the folic acid. So when this folic acid is found in inactive form, it cannot be in this form, it cannot be utilized. Hence, a cancer cell, which is mostly a highly repetitively replicating cell, they require more amount of nucleic acid than a normal cell. So they require more amount of folate for survival. So in absence of folate, these highly replicating cells get damaged first. So first comes cancer cells, second comes cells which are normally have very high turnover rate. So for example, hair follicular cells, normally blood cell producing cells, and intestinal mucosal cells. So these three cell lines, they are affected if we give any anti-cancer drug to a cancer patient or a normal patient. In addition to cancer cells, these three cell lines also get affected. So a cancer patient, they not only get rid of cancer cells by addition of this methotrexate, they also get rid of normal hair follicle producing cells, normal blood cell producing cells. So they suffer from anemia. So they suffer from hair loss. So they suffer from malabsorption syndrome because mucosal cells are turned over rapidly. Right? So all these things happens because of this reason, the cancer patients are always supplemented IV through intravenous route. Orally, if you give vitamins, they cannot absorb because of this problem. Sit down, sit down. Now take down this particular thing. This particular picture contains almost all the reactions of folic acid. At the center is a tetrahydrofolate pool. This pool has several arrows which are utilizing folate and few arrows which are replenishing folate. Right? This is one reaction which is replenishing folate. Another reaction is this reaction, see. One is from folate directly, tetrahydrofolate, and is from N5-methyl tetrahydrofolate. Clear? So these two reactions are important to replenish the folate pool inside the cell. Clear? What are these reactions which utilize tetrahydrofolate? In different form. It can form polyglutamate, that is a storage form of folic acid. It can form N10-formyl tetrahydrofolate, which gets converted to N5-N10-methinyl, and from there it can form this one, or it can take this route to folate. It can form N5-formyl, then from there it can form N5-N10-methinyl, methyl, N5-N10-methylene, methinyl, methylene, and then methyl. Methinyl, methylene, this is methylene, and then methyl. Then it comes back to this. Now, in our body, there are only two reactions which converts N5-methyl tetrahydrofolate to folate. Only two. One of the reaction has been shown here. What is that? Homocysteine to methionine. Another reaction is formed conversion of propionyl-CoA to succinyl-CoA. Propionyl-CoA to dimethylmalonyl-CoA. Dimethylmalonyl-CoA to L-methylmalonyl-CoA. L-methylmalonyl-CoA to succinyl-CoA. This L-methylmalonyl-CoA to succinyl-CoA requires one isomerase enzyme and N5-methyl tetrahydrofolate as coenzyme. So there are only two reactions in our body which utilize N5-methyl tetrahydrofolate, and so there are only two reactions in our body which converts N5-methyl tetrahydrofolate to folate, and these particular reactions are very important because it can produce folate back from its different other forms. There are no other reactions which can form folate back, because once the folate is formed, from folate again all other forms can be synthesized. So the same folate can be cyclically used. So to complete the cycle, we require these reactions. And remember, these reactions, both the reactions, this homocysteine to methionine and L-methylmalonyl-CoA to succinyl-CoA, these two reactions, both the reactions require methyl cobalamin as coenzyme also. That means in vitamin B12 is necessary for reproduction of folate. Vitamin B12 is necessary for reproduction of folate. Hence, a deficiency, a primary deficiency of B12 may lead to secondary deficiency of folic acid or false pseudo deficiency of folic acid, because folic acid is present but in different other forms which cannot be converted to this form, this form back, because all other forms will come to this form. All roads will end at N5 methyl tetrahydrofolate. Try to understand very clearly. All the routes, methylene, methinyl, formyl, formimino, all must end at N5 methyl tetrahydrofolate. From there it must convert to folate, and then from folate it must convert to other forms. Correct? So the major conversion of folic acid rely on availability of B12, and deficiency of B12, a primary deficiency of B12 may give rise to secondary or pseudo deficiency of folic acid. Your body is having folic acid but not in the form of free folate. It's all folate is found in the form of N5 methyl tetrahydrofolate. Other forms are not available, whereas other forms are also equally required for all reactions to happen. That's why it is called a folate trap. So what is folate trap? Folate trap is a pseudo deficiency of folic acid due to primary deficiency of vitamin B12 or methyl cobalamin, more precisely. Methyl cobalamin. Right? Now, when we have a B12 program, so this is folate trap, you understood, right? And the importance of N5 methyl tetrahydrofolate to folate conversion also you understood. These are the points, these are the areas from where they will ask you question. All these different forms, these are all reactions, arrows. These are all reactions. Multiple reactions are there. We'll see all the reactions one by one. All these reactions, they are involved in transfer of single carbon atom. That's why all these reactions together are called one carbon metabolism. One carbon metabolism or single carbon metabolism. So when if it is asked in this way, what is one carbon metabolism? Explain or describe or something like that. You must mention this part. That's all. Now this part is very complicated. It will be difficult for many of you to remember. Right? For those who cannot put more tags on the upper half, for them there is an easier one. Right? The other easier one to remember, one carbon metabolism. Only thing is here, from here to here, you must draw a line. You must draw a line in order to explain folate trap. In order to explain folate trap. If you want to explain folate trap with this diagram, then you have to add one more line from N5 methyl tetrahydrofolate to formate mentioning that particular step where methyl cobalamin requires as a coenzyme. Alright. So these are the easier way to remember one carbon metabolism. Clear? Here I have mentioned all the reactions where these forms are required. N5-N10 methylene tetrahydrofolate required is serine hydroxymethyl transferase and thymidylase synthetase. Okay. You can write this name and the name of the enzymes. These two columns you write. These two columns you write. The third column just explains the same reaction. Doesn't matter. These two columns only you write. N5 and right, N5-N10 methylene tetrahydrofolate. The reactions require this one as coenzyme is serine hydroxymethyl transferase and thiamidylate synthetase. Serine hydroxymethyl transferase is an enzyme which converts serine to glycine, and thiamidylate synthetase is an enzyme which synthesizes DTMP from DUMP. N10 formyl. Most these all reactions are from purine nucleotide biosynthetic pathway. This has got another importance. During amino acid metabolism, have you come across the test called FIGLU test? FIGLU excretion test? Have you come across? Anyone? Formimino-glutamate excretion test. It is a test done to assess folate deficiency. It is a test done to assess folate deficiency. Right? The patient is fed with load of histidine, and if the urine contains high amount of FIGLU, then that is due to absence of or deficiency of folate. So this is that FIGLU excretion test step. Right? See, histidine, urocanate, FIGLU, glutamate, THF to 5-formamino, THF is called histidine load test, done to assess folate deficiency. এটা হয়ে গেছে? ছোট্ট করে লিখে নাও, make it small but write it, it's important. So দুখানা জিনিস শিখলাম, একটা হচ্ছে folate trap, আরেকটা হচ্ছে FIGLU excretion test. এগুলি পড়াচ্ছি না, দরকার নেই। এই রিঅ্যাকশনটা লেখো, এটা। This is important for both, for B12 as well as folate. Role of folate as coenzyme, role of B12 as coenzyme, role of B12 in folate trap, or what is folate trap? One reaction. One reaction, 4 questions. Very few I am just projecting from the whole chapter, but I would expect you, all of you, to remember these steps. I am ruling out all the steps except few. Those few steps you must remember. When I am emphasizing, remember this slide, these steps, that is important, you have to. Which is not required, I will let you know. But what is required, you must not forget. Cobalamin, N5-methyl tetrahydrofolate plus cobalamin forms methyl cobalamin. Methyl cobalamin donates its methyl group to homocysteine. Homocysteine becomes methionine. Methionine then undergoes metabolic reactions, catabolic pathway of methionine. The first one is the formation of SAM. Second one is the formation of S-adenosine homocysteine. Then from S-adenosine homocysteine to homocysteine. Then from homocysteine to again methionine. And from this side, one side it goes for regeneration of methionine, but the other side it goes for synthesis of what? Homocysteine থেকে নিচে যায়, কোথায় যায়? Catabolic pathway methionine কি তৈরি করে? অ্যাসিড টু অ্যাসিটেট, তাই না? আর ফিউমারেট, ফিউমারাইল অ্যাসিড টু অ্যাসিটেট হলো। নেক্সট deficiency of folate. এবার দেখো ব্যাপারটা বলি, the patient comes with folate deficiency. What he presents with? Correct? Severe anemia. Why? Have you given any because of this is happening? So take the history. Whether he has been given any anti-folate agent as drug, whether he is having pregnancy period, correct? Whether the newborn has born with birth defects. So must be the mother had folate deficiency. Some drugs which utilizes folate, or cancer. Why cancer causes folate deficiency? Because cancer utilizes all the folic acid for synthesis of nucleic acid. So what happens? Our body requirement of folic acid increases. Hence there is folate deficiency in cancer patients. What are defects? Very, very important. Spinal bifida, bifurcation of the spinal cord of newborn. The neural tube development is defective. Spinal bifida is one of the condition. Another condition is microcephaly or anencephaly. Microcephaly means the head is very small compared to the whole body. No brain at all. The portion vault is very small. There is no space for brain because brain is not developed. Correct? Anencephaly means no brain. From here it is flat. This dome shape is absent. From here it is flat. Or mostly they die. They don't survive. Microcephaly, they survive with neuronal degeneration diseases. Obviously. Okay. Next is megaloblastic anemia. Megaloblastic anemia I will not tell here because already it has been told in the physiology. Now, what are the drugs that are called folate antagonists? You may be asked a question from this point of view. A patient taking this and this in such and such or in following diseases. Which of the following diseases will contain, well, the patient will have such and such drugs? 5-FU, 5-FU or 5-fluorouracil is a potent anti-cancer drug. Methotrexate is a potent anti-cancer drug, but these are also used in certain benign condition, in certain psoriasis and BPH, benign prostate hypertrophy. Correct? In certain benign condition also, these drugs can be used. Certain psoriasis and BPH, benign prostatic hypertrophy. Okay? So these are the diseases or conditions, clinical conditions where these drugs can be used. Cobalamin or vitamin B12. Vitamin B12 is found in 4 forms, 4 existing forms. Cyanocobalamin, hydroxycobalamin, methylcobalamin and 5'-adenosyl cobalamin or 5'-deoxyadenosyl cobalamin. Now, what are the uses of these four types of cobalamin? Cyanocobalamin is usually found from plants. It's a storage form of cobalamin in plants. Hydroxycobalamin is a water-soluble injectable form of vitamin B12. 5'-deoxyadenosyl cobalamin এটা গন্ডগোলের। Now this is actually the active form of coenzyme. This is active form of coenzyme. N5-methyl tetrahydrofolate, methyl cobalamin is a storage form of B12 in animals. Right? Now you don't ask me why, I don't know. Because all the reactions I have explained are mostly methyl cobalamin. But while telling, I am telling adenosine cobalamin is a coenzyme form. Why, I don't know. Okay. What is central metal ion? Cobalt. What is the ring structure called? Anyone? Corine. Hexafluorescence top আছে, দেখ এখানে দুটো রিং একসঙ্গে জোড়া আছে, এখানে মাঝখানে আমার এখানে ব্রিজ আছে, এখানে কোনো ব্রিজ নেই, ঠিক আছে, 1 আর 4 এর মধ্যে ডাইরেক্ট কানেকশন আছে। এটা ডিফারেন্স বিটুইন কোরিন রিং অ্যান্ড টেট্রাপাইরাল রিং, হিম রিং আছে, হিমের সঙ্গে, ঠিক আছে। হলো, তাহলে ইট কন্টেইনস আ কোরিন রিং, সেন্ট্রাল মেটাল আয়ন ইজ কোবাল্ট, কোরিন, CORRIN। কোথায় পাওয়া যায়? বলা হয় যে ভেগানস আর ডেফিসিয়েন্ট ইন ভিটামিন B12। মানে যারা ভেজিটেবল খায়, ডিম, দুধ ইত্যাদি খায় না, ডেয়ারি প্রোডাক্টস অ্যান্ড মিল্ক প্রোডাক্টস they don't eat, eggs they don't eat, vegans, strict vegans, they suffer from B12 deficiency. They suffer from B12 deficiency. But if the person is vegetarian and he takes dairy products, mostly fermented dairy products, they don't have this problem. If you are vegetarian and if you are taking curd, paneer, or any kind of fermented dairy products, because that contains yeast which produces B12, and yeast becomes our source for B12 for vegetarians. Otherwise, for those who are non-veg lovers, they can eat because B12 is found from non-veg source mostly. Again, most of the functions of B12 are for cell growth maintenance, and that is through folic acid. B12 alone cannot do anything. Through folic acid, they pursue their functions. Through folic acid. Okay? Is there, what is pernicious anemia? Microcytic anemia, you know. Hypochromic anemia, you know. Megaloblastic anemia, you know or not? Yes or no? বল, হ্যাঁ, পড়ানো হয়নি? গুড বড়া। মেগালোব্লাস্টিক অ্যানিমিয়া কাকে বলে? দাঁড়িয়ে কথা বল। কি বল? মেগালোব্লাস্টিক অ্যানিমিয়া কাকে বলে? হ্যাঁ, ডিফেক্টিং আরবিসি। কান্দ যাই যাক, কি বল? বোন ম্যারো, তুই যাই যাক। কি বল দাঁড়া? Megaloblastic anemia means what? এই শুধু অবস্থা, তোরা কি করে হায়ার সেকেন্ডারি পাস করলি? কি বল? হ্যাঁ, চশমা বল। হাব ভাব তো এমন মনে সারাদিন পড়াশোনা করছিস, বল এবার তুমি। দেখো, চশমা খুলে সানগ্লাস পর। কি বল? কে পারবে? এনিওয়ান? কি মাইক্রোসাইটিক অ্যানিমিয়া তুই আর বলিস না থাক megaloblastic anemia means they don't contain erythrocyte, they contains erythroblast, not unmature, immature erythroblasts which contains nucleus still, because we know mature RBC has no nucleus, but megaloblastic their size is big but they contain nucleus. So the RBC, when there's bigger in size containing nucleus, that is a blast form, they are called megaloblastic anemia. It usually occurs when the cell synthesis and DNA synthesis has got a disparity. The speed is not equal. The cell is synthesized at higher rate and DNA synthesized at lower rate. So by the time DNA is synthesized, cell becomes larger in size. And it come out of the blood producing tissue to the circulation. So circulation contains more and more blast form of pro-erythroblasts. Right? So they are called megaloblastic anemia. Microcytic means the size of the RBC is small, smaller than 7.2 micron width. So that means they are smaller. Right? Hypochromic means the iron is less. Here pernicious anemia is something different. What is the finding in pernicious anemia? The pernicious anemia, the blood slide shows megaloblastic cells. In pernicious anemia, the blood slide shows megaloblastic cells, but there is no trace of folate deficiency. Number one. Number two, no trace of iron deficiency. That means what? What is wrong? That means B12 is somewhat might be deficient. But how to find how B12 is deficient in the body? For that, to find out the deficiency of B12, we need to know how B12 is absorbed. How B12 is absorbed? বল কে পারবে পারবি হ্যাঁ বল Cassell's, there are two factors, Cassell's intrinsic factor, Cassell's extrinsic factor. Cassell's intrinsic factors are secreted from the stomach cells, which binds with Cassell's extrinsic factor, which is nothing but B12, and they together they are absorbed. Now, in case of gastric carcinoma, the production of Cassell's intrinsic factor is markedly reduced. So, even though the food, the diet contains good amount of B12, our body cannot absorb it. So, body suffers from B12 deficiency. So, from where we should find out? We should aspirate the gastric juice and test for presence of Cassell's intrinsic factor in that gastric juice. Stomacher juice ke aspirate korte hobe, ki diye aspirate korbo? By using Ryle's tube. By using Ryle's tube. And then we must assess, we must find out whether Cassell's intrinsic factor is present in the juice or not. If it is present, fine and good. If it is not, then probably there is a reason why the patient is having anemia. In most of the cases, it is found it is a cause of this decrease in CIF, nothing but cancer. Okay? বসুন Sit down. Another side effect of B12 deficiency is demyelination. What is demyelination? What is myelin sheath? Very well. What is myelin sheath? Joyful. Louder. Which covers the nerve. Yeah, as in তুই একটা রিক্সা বলছিস। একটা শিক্ষিত তুই একটা ডাক্তার তুই এরকম বলবি? বল। অ্যাকশনস অফ নিউরন কভার করে রাখে। কোথায়? সিএনএস এর মধ্যে না সিএনএস এর বাইরে? ওইটা না বললে সব গন্ডগোল। Outside CNS, outside central nervous system, the nerve fibers are insulated by a thick layer of fat or fatty substance, a lipid substance. It's called myelin sheath. The myelin sheath mostly contains what? Sphingomyelin. The sphingomyelin is secreted, synthesized and secreted by Schwann cells. By Schwann cells. Clear? Now, this myelination is required for propagation of nerve impulse. Say, for example, if when you switch on there and these wire and the switch light is on. If we find these wirings are covered by plastic or rubber or latex, correct? That means insulated. If it were uninsulated, what would have happened? During the passage of electricity, it would have lost a part of it. Because the distance is very less here, only 10 meters, the voltage drop would have been minimal. But imagine a power line coming from Bokreshwar thermal plant to here, the drop would have been very high in voltage. That's why the wire should be insulated for the transport of the impulse from one place here to tip of the toe. Such a long area, right? But it occurs only outside CNS. Why? In CNS, it is already covered by 3 layers. What are they? Not my part of tea. So 3 layers of the brain covering. What are they? Correct. Outside CNS, these matters are not present. It's present only myelin sheath. Now, this myelin synthesis require vitamin B12. In many of the cases, it has been found that the tingling effect or the pain, excruciating pain in the joints are felt very badly because of the open nerve endings. To cover them or to subside the pain, mostly they are given vitamin B12, so that the tingling effect of the nerve is reduced. Right? So one of them is demyelination. Is caused due to deficiency of B12. So 3 major deficiency disorders. One is pernicious anemia. Again, it's a side effect of mostly gastric carcinoma. Neuropsychiatric disorder, mostly B12 deficiency, mostly due to some other reasons. Chromosome damage, correct? Mostly due to some other reasons. These reasons, mostly radiation, mutation. Radioactive B12 can be used for diagnosis of pernicious anemia. How? হোয়াট ইজ দ্য রুট অফ অ্যাবজর্পশন? দাঁড়া না, চেষ্টা কর না একটু, সবকিছু আমি বলবো, হ্যাঁ স্ট্যান্ড আপ, চেষ্টা কর। আমি বলছিলাম এইটা দেখ, এখানে দেখাই দেওয়া আছে, এটা কি করে বুঝবো? আমি একটা হেভি ডোজ খাওয়ালাম B12 ওরালি, তাই না? এবার ব্লাডে মাপবো, ইফ it is absorbed from the stomach, it will appear in your blood, then it will appear in the urine. If the urine doesn't contain, that means it is not in blood, it is not absorbed, তাই না? তাহলে এর থেকে বুঝতে পারবো যে অ্যাবজর্পশনের প্রবলেম হচ্ছে B12 এর।